I'm starting the record.
Yeah.
So actually, this is our first talk in our seminar AISMBR, which is Artificial Intelligence and Statistical Mechanics in Biomedical Research. So we are delighted to have Dr. Dubey as our first speaker for AISMBR webinar series. And he is assistant professor in Department of Chemistry at Nadar University. And he's an expert on cytochrome P450 enzyme, QM and MM analysis, MD simulation, and the docking. His research involves proteins and protein ligand interaction, enzyme engineering, and computational drug discovery. He's the author of several research papers in a prestigious journal like Nature Communication, JACS, Catalyst, a lot. And throughout his academic career, he uh, he received a prestigious national and international fellowship from the Sweden and the DBT. And currently, he is also serving as a reputed journals editor like PLOS1 and the Frontier Chemistry. And we're very happy to have you, sir. Now, floor is yours. And I will request audience, you can put your question in the chat, or at the end, also you can raise your hand.
Um, okay, so I'm sharing my screen. Whether is it visible?
Yes, sir, it's visible.
Okay, good. So thank you, Dr. Pratiti, for this introduction. So today I'm going to talk about cytochrome P450, and basically it is a type of new insight. And I will try myself to refrain from some technical and much chemistry. So it is a type of philosophical talk where actually we are providing a new insight about the enzymes. So whatever the work today I'm going to present, so that was done in collaboration with Professor Sosan. So I am feeling that he is imaginary, I will say that speaker here, because all those work we are done in the collaboration with Sosan. So let us start with some philosophical things. So we know that something like 3.5 billion years back, the entire world was abiotic. We had no any life. And now we have very complex system like biological system. And now we are living in biotic worlds. There is very great chance that we are rapidly evolving and might be after 50 to 100 years, the entire worlds from abiotic to biotic, we are going to be a robotic worlds. Most of the times, most of the things in our worlds, in the surrounding, you will see that robots are moving, and most of the things will be done by the artificial intelligence and the robot. Even there is a very recent publication in Nature where using artificial intelligence, actually people found that the AI predicted almost three-dimensional structure of all protein universe. And they found that those structure which was predicted by artificial intelligence, that was very accurate, even at level of the experimental structures. So you can see that now artificial intelligence, robotic things, they are very, I will say that um, nicely increasing, and their application is also increasing. Even just by doing some type of search in SciFinder, you can see that the number of publications increasing using computational chemistry keywords. And that was completely, I will say that it was lacking something like before 50 years back. But you can see that there is a sudden rise in the number of publications using computational chemistry. Now the question is that what actually this computational chemistry is doing, why these people are very much interested in this computational chemistry, why all type of people, even the experimentalist, mathematician, everyone now doing some type of computational things. The answer is that it is actually the insight, because it is providing us a very nice insight about the mechanisms, about the functions, not only in the chemistry, but all those physical phenomena, even the biological phenomena everywhere. So let us give one example that how insight matters. Just for example, you know that the gravitational law given by Newton in something like 17th century. And you know that actually there was some story that he was sitting in some garden and suddenly he saw falling apple. And then after actually that gave him motivation to think about the gravitational law. But the thing is that whether means that he introduced gravitational law something like 17th century. So the question is that, does it mean gravitational law was absent before Newton? Answer is no. You know that actually the gravity was basically present even after the first minute of the Big Bang. As universe is started, gravitational law was gravity was there. It was basically the insight of the Newton. Actually, the many people were also watching falling of the apple, but it was the Newton who gave the insight, and then actually it led him to discover a new law, that is the gravitational law. So that's why different examples, there are the different people, they have the different insights, and many times new insights give you a very new discovery in the science. I will give my example also. As I am a physicist, I did my PhD, my master's, everything in physics. And then currently I'm in chemistry, I'm working in chemistry. And most of the time I had a slightly different insight from the chemist, because I believe that the knowledge sometimes gives you a constant. If you are reading, you are expert in one area, most of the time you think about the same area. But at the same time, if some people who is also expert, but he is from the different area, he may give you a new insight, and might be you are lacking this insight because you have a knowledge of your specified domain. And that's why always insight is different. So today's story, whatever I'm going to talk, that is a different insight. The reason is that people think about the enzymes at the biological machines. They are doing some type of catalysis. But here actually we are providing a new insight that these enzymes can be treated as the nano machines of the living matters. And whatever the story I'm going to tell, that is basically our several year experience in molecular dynamic simulations of cytochrome P450. This molecular dynamic simulation is very powerful computational tools, and we can study about the conformational movement with simulation of the time, with the course of the time. And I'm not going to tell the technical details of the molecular dynamic simulation, just like using this simulation, what insight we get, I'm going to explain about that. So here I will highlight the some fascinating and very, I will say that autonomous choreography that this enzyme cytochrome 50 is doing during its catalytic cycle. Since I am talking about the cytochrome P450, I believe that it is necessary to introduce this cytochrome P450 very briefly here. So briefly, this P450 is nature's a machine. I will say that it is a type of universal machine. And most of the time, most of the catalytic process is in our body or any living materials that is done by this cytochrome P450. You can see that actually this cytochrome P450 can do plethora of the reactions, starting from alcohol, aldehyde, ethers, nitriles, means that they can metabolize something like 80% of the reaction in our body. Whatever the oral drug we are taking, so cytochrome 50 is that enzyme which actually metabolizes. And that's why it is very important for the chemist as well as the drug developer and the biochemist. And this is the reason why most of the people who are working the drug discovery and then even the enzyme catalysis, so cytochrome P450 gives them a very standard model to study these things. So the reaction is that like it uses the molecular oxygen and then it reduces it and it oxidizes in different substrate. You can see that like it does some hydroxylation. If you have like it does aldehyde formation, it does epoxidation, and every time basically it is inserting one oxygen into the substrate and next oxygen actually converts into the water. So we can believe it is a factory where this is the core machine, and like it utilizes protein and the water molecules. So we can think that these water molecules and proteins are some type of workers. And actually it takes electrons from some reducing agent, and also it uses molecular oxygen. So this molecular oxygen and the electron can be treated as the fuel for this factory. And we insert substrate and then we get oxidized product. So this is a type of a system, I will say that schematic diagram that how this cytochrome P450 can work as a machine. So I'm going to talk about this P450 machines. So the thing is that in this story, what today I'm going to talk about, I am basically proposing that this P450 is a living matter and which has a three-dimensional structure, but it creates four-dimension machines. Now the question is that what is the three dimensions? What is the four dimensions? So we know that all proteins are crystal structures. They have some three-dimensional structure, XYZ coordinate. But this P450 is actually work as a robot, means that it can sense external stimuli and based on that actually it reacts. The reason is that it is a almost dead enzyme which do not work. It is almost silent. But whenever actually it senses the entry of the substrate, then the catalytic engine starts. So it's mean that this sensing is its fourth dimension. And this sensing works in some type of van der Waals interactions and some electrostatic type interactions. And then it does in automated way. Once substrate enters, there are several catalytic steps that automatically it goes on, and then it works in a very synchronized manner, and then it leaves the substrate. So that's why we can say that this P450 are 3D materials with four-dimensional property. Wherefore, again, I am emphasizing the fourth dimension is its sensing property. It can sense the external stimuli. Now, the thing is that this is the structure. In this structure, we can see that it has a core. That core can be treated as an engine. And this core is actually known as the compound one. You can see that there is a ring that is a porphyrin ring, and it is ligated with one amino acids that is the cysteine. And you can see that there are several strategic amino acids, and they are basically naturally evolved around this porphyrin ring. Here you can see that there are some amino acids, just for example, this glutamate and this threonine, and they are basically, people has found that they play a very important role in OO activation. And then there is some amino acids, just for example, this glutamine. So it holds substrate and it provides substrate binding. Then again, you can see it has a propionate side chain, and this propionate side chain works as a gatekeeper. And just by flipping its hand, you can see that there are two propionate, one is here and another is here. Just by making it up and down, it can basically regulate the water channel. It can work as a gatekeeper. Similarly, you can see that there is a phenylalanine. This phenylalanine actually work as the selectivity. Whatever people want to get, like this enzyme, if one wants as reaction as last end, and then omega -1 end, omega -2 end. So basically, it was done by this phenylalanine. People has done several experimental study, and they found that actually by mutating this phenylalanine, the selectivity changes significantly. It's mean this is playing a very crucial. Similarly, there was a person in Japan, Hayashi. He basically removed this propionate group, and he found that after removing this propionate group, these enzymes become inactive. It's mean that although this is the core where iron is sitting, but each component of this heme group plays a very crucial. So what lesson we are actually learning from this one, that nature has installed each and every amino acids with some strategic role, and this porphyrin ring is sitting in the core, and it is basically surrounded by some very tight spacing, and enzyme senses all these external stimuli with help of this tight spacing. So now I will talk about this catalytic cycle, and keep I will refrain to discuss more details about this catalytic cycle. My only motive is that I can say that you can see that in this catalytic cycle, there are several sequential steps. Just for example, one substrate is enters here, it replaces this HOH water, and then it does first reduction, then there will be dioxygen diffusion, then there will be second reduction, then first protonation, second protonation, and then actually it forms one active oxidant. And this active oxidant is basically responsible for the all type of the reactions like oxidation reactions. My motive was that to show that this catalytic cycle or this machine works in a many synchronized steps. And once this substrate enters, everything goes in a very synchronized and very autonomous way. So we will discuss about these things in the later on of these cycles. So the thing is that, like as I'm saying that we can believe this cytochrome P450 not only as a machine, but I and Session we discussed that we can name them as a soft robot. The difference between the machines and the robot is that machine can work in a synchronized way, but it lacks the sense like sensing machinery from its external stimuli. But in this case, cytochrome P450, basically it can change its way of the interaction, its way of its, I will say the activity after sensing the external stimuli. Just for example, if you are providing a very accurate or even the specified substrate, only it will activate. But at the same time, if you are changing the substrate, which is bulky, which is not fitting to this enzyme, it will not work. It means that it is a property to sense the external stimuli. And that's why it is better say that they are soft robots instead of the machines. So here the thing is that this robots works in a different way. Just for example, I'm citing some of them. For example, the protein, which is basically opened when substrate enters, it goes to the closed state. And then just as I have shown that initially when substrate enters, it displaces water. And then again, this water must return so that it can initiate the second protonation and then first protonation, and it can form the active oxidant. And finally, once substrate is oxidized, the cavity reopens and it allows substrate to escape. The reason is that once your substrate is being oxidized, and if it is not going out, the catalytic turnover will stop. Means that there must be very synchronized motion that initially substrate must enter and with course of time after oxidation, this must also exit. So you can see that actually in this enzyme, this soft robots does a lot of motion and all these motions are synchronized. So in this next, I will say slides, I am discussing about such type of movement. So you can see that these completely autonomous functions of this enzyme and this sensing actually it is coming because of the interactions, van der Waals interaction. Here in the terms of the robots or even the machinery, we will say them as a tight spaces. And every molecule of our enzyme, they basically have some van der Waals radii. And these van der Waals radii are basically filling the gap of the protein. And one protein and the protein molecules interacts with other residue using this tight spaces that is the van der Waals radii. So we will be discussing all these things in the next slides. So let me elaborate here. Since I'm saying that this is a robot, so definitely it will have some core. And as I have mentioned that this P450, that porphyrin ring is basically the core, which is the basically the engine of this robot. And you can see that such type of this sausage type of channels. So these are the channel which we can say then them as a water channel, might be substrate channels. So you can see that actually these channel which are connected with this core, they are basically the sensors. And using these sensors, basically this enzyme can sense the external stimuli. And like all these substrate entry, water oxidation, like exchange of the water, removal of the substrate, all these are done by these channels. Okay. Now the thing is that let us see what actually the meaning of this sensing. So as we can see that this protein senses the substrate and just by meaning of the van der Waals interactions and applying some type of weak interactions. And after this again, like interactions, they recoil and they activate the movement of other amino acids they open the gate, and actually they do other several things. Now the thing is that since I'm talking about as a robot, so definitely here actually we introduce one term, that is the soft gearing motion. So soft gearing motion is that, suppose that if we have one amino acids and it is very closely surrounded by the another amino acids. So definitely it has some type of van der Waals radii, and this van der Waals radii or van der Waals surfaces actually interact with the other surface, and by movement of one part, it actually triggers the motion of the another part. So that's why we think that this sensing is a mechanical soft gearing motion. And because of this such type of interactions within the amino acids. And we can say that because of the nature of these interactions, it has some type of sensing mechanism, and using simulations like molecular dynamic simulations. So we have investigated a lot of such type of soft robotic movement in these enzymes. Okay. So now I'm going with each and every step of this catalytic cycle, and then I will discuss that how this sensing and this soft robotic gears are actually synchronized, and they transfer one interaction to the another one. Means that from if they complete one step, then they transfer this property to the another step, and then every step is synchronized, and then ultimately it does such type of product. Okay. So now I am starting with the first step, when substrate binds to the initial state, that is the resting state. So in this case, I am just taking one example of one substrate, that is the N-palmitoglycine acid. Basically, this is a fatty acid, and we can see that its one end is basically the acidic end, and the end is basically the hydrophobic. So what happens in the mouth of this substrate, or sorry, enzyme, there are different charged amino acids. And these charged amino acids applies some electrostatic interaction with this acidic group. And because of this electrostatic interaction, basically they pull substrate inside the cavity and they holds there. And basically these interactions are because of the arginine, this glutamate, leucine, glutamic acid, and tyrosine. And each one of these are a type of polar or charged amino acids, and nature has evolved these amino acids just because of this acidic group of this substrate. Later on, what's happened when actually this substrate enters, so this enzymes open its mouth so that it can very easily accommodate this bulky fatty acids. Now the thing is that you can see we did some simulation, and we found that actually when there is no substrate, means substrate is somewhere outside the solvated box. So we can see that actually these beta seeds, this is basically the mouth of the channel, it further opens. And when actually it further opens, means that it is widening its mouth so that it can very easily accept the entry of the substrate. In the next step, what happens when you can see that when substrate enters here, the same secondary structure which were actually opening in the presence of the substrate, now they are moving inside by 11 angstrom, so that once substrate has entered inside the cavity, now it can block its further opening, and it can hold it very tight so that our engine can perform some specific reaction. And all these things were again done by some, we can say, strategic amino acids, just for example, proline 25. Now the thing is that when this substrate enters, what next happens? So for that, actually, we have plotted some type of water channel parts, some type of cavity and water channel, how water channel is behaving when substrate binds and when substrate not bind. So in this case, this is just a structure of the substrate free state, means if there is no substrate state. We can see there are two parts. One is the distal part, means that the top of our porphyrin ring, and then there is a proximal part, the bottom of this porphyrin ring. So now we can see that when substrate is not there, so these sausage type of tube type of things, what actually structure you are seeing in the screen, they are basically the water aqueduct. So now we can see that there are two major water aqueduct when substrate is not there. It's mean that this water channel is very, I will say that uh, significantly, or it is very predominant in the case of in the upper side, that is the distal side, when substrate is not there. And then again we plot it when substrate is there. So when we again we have drawn such type of structure, this water channel, when substrate is there, now we can see that this water channel, that is the aqueduct, now they are shifting towards the downside, that is the proximal side. The reason is that when substrate is entering here, basically it is occupying the distal shape, and now it is pulling the water behind that one, that is the towards the propionate side. And now here, because of water is now coming to this propionate side, now the role of propionates comes, and it can get close and it can open, it can regulate the water entry after this substrate is coming. So we can see that even like our how this porphyrin or enzyme is basically sensing the presence of the substrate. When substrate is not there, water is more populated in the distal side, and when substrate is there, now it is very much populated towards the proximal side. And all these things are done by some van der Waals interactions. Just because of this, actually, it is repelling the water from one side to another side. Now, the thing is that how this mechanism is basically happening. We can see that actually when substrate is coming, it is basically pulling the water from one side to another side. So we have plotted it as a soft gear. Just for example, whatever structure you are seeing here, so they are basically the van der Waals radii. So we can see that this is the van der Waals surface of the porphyrin, and then there is a lysine residue, and then there is a phenylalanine. And now it is very clearly seen that these surfaces are very close at very close proximity, and just movement of any of these amino acids can affect the position of the other one. And that's why we are saying that it is a soft gear. Just for example, if this gear is changing, so definitely it can change the location of this gear. And in that way, by this movement, it can basically regulate the water channel. So in the absence of substrate, means there is no substrate. So now we can see that this propionate hand of this heme is at the top side, means that at the distal side. And because of this distal side, actually it is opening, and we can again see that this phenylalanine is basically at the perpendicular plane. And since it is a perpendicular plane, there are many water which can come to this side, and it can occupy the catalytic center of our heme iron. But at the time when substrate enters, like this is the NPG, when actually it enters, it applies one soft gear type of force on this phenylalanine. And now we can see that in the absence of the substrate, it was in the perpendicular plane, and now it becomes to the parallel plane. And because of this parallel plane, now it is again pushing this lysine, which this lysine was basically at this position. Now we can see that it is now at the down position, and this hand up with this propionate. Now you can see that initially in the absence of the substrate, it was at the distal side. Now it twisted to the proximal side. And because of this twisting, basically it is blocking the water access. And again, this phenylalanine is also blocking the water access. And this is the reason why substrate is displaces. And actually there is no any such water on this catalytic side. So now you can see that this sensing is basically, or even this trigger of this movement is basically initiated by the substrate entry. When substrate is entering, it is doing a lot of different movement. And because of the different movement, basically water is going from distal side to proximal side. And now you can see that this propionate chain, we are actually mentioning as a prop 7, they are actually working as a handle. Once it is up and then it is down. And because of this handling mechanism, actually it can trigger the different movement of the water. Now we are going to the second step. the second step I have highlighted here, that once I have shown that when substrate enters, it actually replaces the water. And I have demonstrated that how using the van der Waals interactions, basically it is displacing the water. The next step is that when actually water comes, actually it vacates the distal side, and now this heme changes to the penta-coordinated. And because of this penta-coordinated heme, its reduction potential changes, and now it is very prone to accept one electron from a distal part, that is the reductase domain. So second part is that's reducing. Our ferric iron changes to the ferrous by accepting one electron from a very long distant group, that is reductase domain. So now we are going to discuss that how this movement, the initial movement when substrate came, can actually trigger the electron movement here. Okay. So this question is that since when there is a no substrate, there was no reduction is going. And now the entry of substrate from this side, how actually is triggering. So definitely all this movement is done by the substrate. So now I'm going to the next part. We can see that how actually it is happening. So this is the crystal structure. This is the reductase domain, and this is the domain donor, and this is the acceptor. So in crystal structure, actually, we have found that the distance between this donor and acceptor is very large, near about 19 angstrom. And this 19 angstrom, people has actually calculated using the Marcus theory that this distance is so much that it will take some year to travel one electron from here. Means that this distance was not physiological relevant for the electron transfer. Now the question is that then how this electron transfer is coming. And also this is very good news that this distance is very large. The reason is that if it was might be very close, then in that case, even without the entry of the substrate, this might be reducing the heme, and this heme is basically being reduced without any output, and it is basically a loss of the enzymatic activity. So it's mean that now we can see that this distance in the next, I will just slide, I will show that how entry of the substrate actually triggers the this distance, and these acceptor and donor, they basically will come very close. And again, here our robot, like smart robots, comes into the play. And we will see that by getting the signal from the substrate, basically this movement of the domain actually starts. So let us think about that. So initially what we did a simulation, MD simulation, when substrate is not there. And we can see that actually we did some microsecond simulation, and we found that the distance is almost something like 22 to 24 angstrom, which is very, very far away. And that's why we can say that in the absence of the substrate, there is no any possibility of the electron transfer. Now, what actually we did, we docked the substrate in the crystal structure, and then we performed the simulation. And now we can see that when substrate is there, now the distance between the acceptor and donor now started decreasing, and they are coming something like 12 angstrom. So we are starting from something like 20 angstrom, and by presence of the, just by due to the presence of the substrate, now we can see that the distance of the acceptor and donor is reducing significantly, and it is coming something like 12 angstrom. And people has done some theoretical study, and they found that this 12 angstrom is biologically relevant distance. Here electrons can move from acceptor to donor. Then actually we did one MD snapshot. We took one MD snapshot, and then we did some QMMM optimization. And we found that after the optimization, this distance is further reducing to 8.5 angstrom. And now using this Marcus theory, we calculated the rate of electron transfer at this 8.5 angstrom. And we found that this value is very nicely agreeing with the experimental KCAT rate of electron transfer. Means that whatever the distance we are getting after our QMM optimization that is basically matching the experimental one. So it's mean we can see that in the absence of the substrate, the distance was far so that there was no electron transfer. And now when substrate is coming, this is reducing. But there is a question that how the entry of the substrate, which was basically this side of this, like this side, is changing the distance or triggering some disturbances. And because of this disturbance, these two amino acids, two acceptor and donor are two groups are coming close. So let us actually see some, I will say, functional helices or secondary structure, which are basically participating. So I have highlighted one helix as a alpha one, and then there is a C helix in a green, and then this I helix of the I helix of this heme domain. So this I helix and the C helix are basically a part of our donor, that is the donor domain. And then this alpha 1 helix is a part of our, this is acceptor domain, and this is from the donor domain. Now we can see that these three helices are at a very close proximity. Okay, so now we will see that how the entry of the substrate is basically doing some type of soft movement or soft frictional movement. And because of this soft frictional movement, our this soft robot is sensing its machine mechanism, and it is actually doing some type of work. So here actually we can see that in the case of substrate free, when there is no substrate, basically this alpha 1 helix and this C helix. Again, let me go here. This is the alpha 1 helix, and this is this is the alpha 1 helix, and this is the C helix. So what happens? You can see that this alpha 1 helix of this FMN and this C helix of heme, they are almost in perpendicular plane. You can see that the distance, the angle is near about 88.2. And in this case, this aspartate is making a very strong hydrogen bond with histidine. And because of this interaction, basically this helix and this helix are basically in a perpendicular shape. But with course of time, this alpha 1 helix is actually it moves. And now we can see that this distance or even the angle between this alpha 1 helix and this C helix are basically changing. And now they are becoming almost, I will say that in the parallel way, because the angle is now from 88.2, it is changing to 31.9. And now we can see that this interaction is basically shifting. Initially, this aspartate is making interaction with histidine. Now it is changing partner, and now it is making interaction with the other part, that is the lysine. And the interaction with histidine is basically weakening. And because of this interaction, actually it is pulling the alpha 1 helix from perpendicular direction to plane to parallel direction. Now there is again a question. The question is that how this movement is basically triggered by the substrate entry, how our soft robot is basically sensing this mechanism. So let us see in this one. Now, when actually we dock the substrate and we perform the MD simulation, now we found that the entry of the substrate is actually creating one kink in the I helix. Again, let me go here. This was the I helix. In absence of the substrate, you can see that it is completely a type of straight rod. But once substrate is entering, now you can see there is a kinking. And because of this kinking, the aspartate of the I helix is now actually interacting with this lysine. You can see that when there is no kink, the distance is 10.8 angstrom. But because of this kinking, now you can see that they are actually in a very good way. So when substrate is actually coming here, it is basically interacting with this lysine. And you can see that this lysine. So means that in the presence of the substrate, now this aspartate is locking this lysine so that now it cannot move. And since it cannot move, so the domain movement is also restricted. And that's why it is now pulling, locking in one condition, and the distance remains at a very close. Okay. So now we can see that clearly this substrate binding provides a driving force that induces extensive motion in the neighboring protein, and propagates thereby the catalytic cycle. Means we are coming, we have seen substrate was entering here, it was opening the gate, and now it was reducing. So let us go to the next step, and again we will see that how substrate entering is now propagating to the third step, that is the O2 binding. Okay. So in that case, actually, we did now the simulation with dioxygen, and we actually found two steps. We found that when there is a initial step, initially this phenylalanine and this substrate, they are basically phenylalanine are it is in the perpendicular plane, and because of this perpendicular plane, basically you can see that it is providing a room to the substrate so that substrate can occupy just the top position from our catalytic point. And in that case, substrate is basically occupying this part. But we found that during the simulation, when the molecular oxygen is entering, actually it is producing one pressure on this phenylalanine. And when this phenylalanine is moving from perpendicular to plane direction, now actually it is pulling the substrate outside. And when this substrate is moving outside, now it is making some room or some space so that this dioxygen can come here. So now you can see that with course of time, initially this dioxygen was completely away from our catalytic site, but because of this movement of the substrate and this phenylalanine, now this dioxygen is entering and now it is occupying the catalytic site. And then we did some QMMM calculation, and we found that the binding of this dioxygen with this iron was almost barrierless, and which was completely agreeing with the experimental rate constant that the binding of this O2 with iron was 10 to the power 7 per second, means it is quite fast, which was almost similar to what actually we got in our QMM calculation. So now you can again see that even this O2 dose, which was provided to our soft robot, that must be very precise, because if it is very large, then it can pull this substrate outside the cavity, and then might be our substrate, our catalytic engine becomes inactive. So again, we can see that basically it is a very synchronized movement of phenylalanine, and it is again triggered by the movement of the substrate. So let us go to the another part. Like we have seen that we have gone from this to this one, then this to this one, and this to this one. Now we are going to this part, like how this part is again synchronized. So let us think about this protonation part, first protonation part and the other, and then ultimate formation of the active oxidant. Okay. So the question is that in the first cycle, we have seen that when substrate enters, it basically displaces the water. Now the question is that if we are going to make the ultimate oxidant, we again need some type of proton. So once this water actually they have leave the pocket. So the question is that how again they are coming so that they can protonate our oxidant to make the compound one. So now we are going to see that. So here actually we found that this propionate side chain, they are actually, this is the secondary water channel. This is basically by the glutamate and threonine. And then there is a primary water channel. And this primary water channel is basically regulated by this propionate side chain. We can see that in the initial case, in the when substrate enters, so we have seen that basically it is completely locked, and it is doubly locked by serine and then this arginine. And because of this locking, now the water from our, I will say that proximal side cannot enter to this distal side, and then it cannot provide any source of the proton. It is completely locked. Now the question is that how again it is coming here. So it is the same like we have seen the entry here. So for that case, actually what we did, we now attach the another protein, that is the reductase protein, with our heme, that is the main catalytic part. We found that whenever the reductase partner is attaching with our heme partner, So now we found that there is another amino acids, that is the methionine 490. And this methionine 490 is now interacting with this serine. And because of this interaction with serine, basically this log, which was, you can see that previously, the serine and the propionate, this distance was 2.7. And now because of this interaction with this methionine and serine, basically this distance is opening. And now it is, and again, our this arginine, which was basically blocking with this propionate, is completely opening. And since this gate is open, now we can see that very sufficient amount of water is now coming at the distal side, and they are occupying the active sites. So again, you can see that just by attachment of the reductase domain, the gate which was blocked after the substrate entry, now it is opening, and our water can enter to the active site so that they can participate in the first and the second protonation step, and they can design such type of active oxidant which can do such type of reactions. Now the thing is that we can see here that the sensing ability of this enzyme is completely restricted cavity. We have very restricted van der Waals cavity, van der Waals surfaces, and these restricted motion are basically leading to the opening of the water gates, and they dictate different routes of the two protonation, and which eventually gives the rise to the active species, that is the compound one. Now there is the last question. The last question is that once this substrate is oxidized, now it has to leave the pocket. For that, actually, what we did, we have done some type of steered MD, and we took this oxidized product. And we found that initially what happens, there are two amino acids, that is the phenylalanine 215 and phenylalanine 57. And now we can see that they are basically the blocking the exit of this substrate. But once actually it is protonated, during the dynamics, we found that the distance, which was initially 4.85, now it is basically changing to the 11.25, means that they are actually now opening. And because of this opening, actually the substrate, the oxidized substrate can exit. And again, they are coming to the very close to the 7.85 angstrom. Again, you can see that this enzyme can very easily sense that when these two amino acids must go apart, so that now it can open the cavity and our oxidized substrate can exit. So it is completely, we can say that it is completely magical way that these weak interactions, because of this van der Waals and electrostatics, even there is no covalent interaction, these weak interactions serve as a role, serves very crucial role in sensing the product molecules, and they direct its departure. So at this point, we have completed the details of entire catalytic cycle. And now let us take some home lesson that actually what we have got the information from these soft robots. The first thing actually what we got, that in this entire catalytic cycle is very complex, and it is progressing automatically in very logical sequence. And it is well timed, that initially when substrate comes, then the channel should open, and then it's closed, then it must displace the water. When water displaces, now the thing is that the another part of the domain, actually it comes, then there will be some shifting of the helical motion. And because of the helical motion, now two domains are coming together. So you can see that they are very timed. It is just like we are making a watch where your second, the gear of your second is connected with the minute, a minute is connected with the hour. And once one gear is working, and once 60 seconds is complete, now it moves to the first minute of the gear of the minute. So now you can see that they are completely working as a timed machine, and looks like the nature has made all these things in a very sophisticated way, such a watchmaker. And now this you can see that in P450, and entire this activity is achieved all these amazing features because of the protein has a very restricted space. And we have seen that propionate is connected with the phenylalanine, phenylalanine is connected with another one, and they all are very restricted motion. And because of this restricted space, they are basically work as a very sensitive sensor. And now we have also seen that this sensing and its response by the protein are basically, they are leading a series of the events, and these events are dictated by different others, I will say that events. And it looks like that enzyme has its own sense, its own feeling that when substrate is entering, when and how other event must be triggered. So in that case, we can see that the sensing and response action of this soft robots with a 4 dimension. Again, I'm saying that this 4th dimension is the sensing of this enzyme, which is basically absent in the regular three-dimensional material. Because if we are designing any 3-dimensional material, that will just have a shape, but it has no any autonomous sensing, and that's actually differs these enzymes with a normal 3D materials. Now the thing is that we are already in an exciting future where soft robotics will play a major role. And now we know that actually there are several 4D materials are now being produced, and they can be printed by like we have already some 3D printing machines, and now these materials, 3D materials can be printed with hydrogels. And using our chemical intuition, we can actually develop 4 dimensions property just by changing the temperature or electric field or changing the humidity. And these property can trigger their sensory mechanism, and which can create the 4 dimensions. And these soft robots machine will provide chemists with a large, I will say that, arsenal of systems that resemble enzymes. And there is a very nice, I will say, the review article by Magdasi, and he has written how these 4D materials like can work in the chemistry as a soft robots. Now the thing is that we are completely in the era of the soft robots, and certainly we are in for some exciting future of the soft robotics. And now the thing is that we need to invent such new self-evolving polymers or gels and to produce from the scratch, so that by triggering some type of 4th dimensions or even the sensing, they can make a perpetual machine. And we have to work in this direction. And even I will say that nowadays, using the directed evolution, one can achieve this goal. People are basically uh, engineering enzymes for the different activity just by knowing that if they are going to change this property, now the chemical reaction will change. And in 2019, Francis Arnold, she got the Nobel Prize for this directed evolution because she can invent a new reaction just by mutating these enzymes. Means in some way, you can program these enzymes by knowing these properties just by tweaking such type of things. Now, all these results were done by us using computer simulations, and these works were done in collaboration with Sasan. And here are some references. So where these data were taken. So we have published, even I will say, very 3 nice review articles, accounts of chemical research, and 2 articles in Trends in Chemistry of the Cell Press. And if you want, one can go. And with this, I'm just finishing my talk. Thank you.
Thank you, Dr. Dubey, for a nice and insightful talk. So I will ask you the first question, then I will go for the audience.
Sure, sure, sure.
Yeah. So actually, I just want to know that the distance, the donor and acceptor distance, or the position of the F87 amino acid, is it parallel or perpendicular?
Yeah, yeah,
Does
yeah,
it
yes.
change in depend on the inhibitor inducer? So enzyme have an inhibitor and inducer. Like, does that conformation change that donor and acceptor distance, and also the F87 conformation? Does it take a different conformations in case of the when the inhibitor pass into or when
Yeah,
into
so people
star
has
pass
done
into.
some yeah, exactly. It is nice question. So basically, people has done crystallographic study and they found that when there is no inhibitor, so the structure of our protein is different. And when substrate is entering there, they have done co-crystal with substrate or inhibitor, and they found that the structure of the protein is different. Even there is experimental structure also that when abstract inhibitor is not there, this phenylalanine has a different position. And in the case when substrate is there, actually it has a different position. So that
Even,
was almost same as what we got.
yeah. So even also inducer means inhibitor inducer to different substrate as per my understanding. So there is a crystal. So the when the inhibitor is bind and inducer is bind, both take the similar structure.
I was saying that when inhibitor is there and when
Inhibitor with the complex enzyme, the
yeah,
conformation of the enzyme, and when
yeah.
inducer and enzyme, conformation of enzyme, these two conformation are same.
I
Because
didn't
the
get this
pump,
induced.
okay, actually, I think the inducer increase activity of an enzyme. Am I right?
Um,
I'm
no,
not.
no. I will say no. The reason is that these enzymes are very specific.
Okay,
Like they are
okay.
so specific that they will work with only one substrate.
Understood. Understood.
Yeah.
Okay. And sir, another thing that you have that is EMD simulation for the distance of acceptor and donor, and also to see the how the helix change or the bond formation of some F87 changes. So do you run only single conformation and then you match with the experiment, or do you run 3 times or 5 times?
No. We actually
Simulation?
we used to simulate different replicas. We does
Yeah.
3 to 4
Okay.
replicas,
Yes. Yes.
and we
That's
found that
actually.
actually they were very much consistent in the all replicas.
All atomic are some course because that 6 Armstrong and 11
Yeah.
Armstrong movement, like
Yeah.
distance
So
6 Armstrong. So is it all atomic?
yeah, all were all atomic simulation.
Okay, thank you, sir. So if anyone have you open your mic and ask, or you can raise your hand.
Yeah, so I have a couple of questions. Can you hear me?
Yes, I can hear.
Yeah. So how do you map the water channels? Is it by the
there is
trajectory of the water?
Yeah, basically there is a one program known as Caver. In that case, we can upload the MD trajectory and then it can make it. So there are two ways we can take different snapshots, like one initial, then middle, and then last. We can also track according to that one. And there is also option that we can upload entire trajectory and this program will give us the this cavity.
Okay. What is the density of water in these channels? Like at a given time, is there only one water? Are there multiple waters
No,
in the channel?
randomly. Yeah, so the thing is that, like, I will say that it was 5 to 6. Like, actually, we plotted a radial distribution function. Like, we just took like active site and then we took 5 angstrom and then we actually plotted the water population. And we found that when water is there, like when there is no substrate, this amount was very small, maybe sometime 1 or sometimes 0. And when substrate is not there, it was 5 to 6. So the population was near about, I will say, 5 to 6 water molecule.
Okay. Okay. And then one about the oxygen, the O2. When the O2 comes in, is it? I mean, is it free O2 or is it bound to a residue before it binds to the heme?
Yeah. So I will explain here. This is actually very nice and very technical things. Actually, it is free O2. But the thing is that if we are going to the free O2, so since you know the DFT, we will not get any charge of this O2. So actually we modeled as a three site O2. like it is a O3, practically it is O3, and we actually compensated the charges such a way that actually its quadrupole moment is replicating a O2. So it was O2, but water model, so oxygen water actually what we took, that is the 3 site, just to replicate their charges.
Okay, but it's kind of more, it is closer to, I mean, the density would be closer to the gas phase O2
Exactly.
rather than
Yes.
a bound
Not
O2.
no, not
Okay.
gas phase, but liquid state. Actually, people have done some benchmarking, and they found that this 3 site O2 is basically replicating the quadrupole moment of the water in the solvent. It is not the free,
Okay,
like in the not the gas phase. Yeah.
so solvated water.
Exactly, salvated water.
Okay, okay. Okay. Okay, thanks.
We have another question. Dr. Diti Chetan or Diti Chetan, can you unmute yourself and ask your question to the speaker? Diti Chetan.
Yes, sir, am I
Yeah.
audible clearly?
Yes, yes, yes.
Sir, my question is that about the physical environment of the enzyme. So, will it affect the, because you mentioned that it is only the presence of the substrate which will affect the opening or closing of the enzyme. But let's say I change the pH conditions or the pressure or um, the temperature that the enzyme and the substrate is present in. Are there any changes that happen? Like,
Yes.
is there a less substrate binding or
Yeah.
anything of that sort?
Yes, this is a nice question. Yes, I will say that definitely. The reason is that these enzymes are, again, I'm saying that they are very specific and they work under certain conditions. Like if we are taking this enzyme as a different pH and like we are changing the pH cases, in that case, the property of enzyme will change. The reason is that if you are going to change the pH, so definitely the protonation of our charged amino acids will change. And if protonation state of our charged amino acids are changing, it's mean that our the electrostatic interaction or the polar environment is changing, and that may change the mechanism of the enzymes. So definitely the conditions of the physical conditions, physiological condition of your surroundings is playing very important role in enzymatic activity.
Thank you, sir.
Anyone have any more question? Type in chat box.
Well, let me congratulate you on a fascinating story that you have mapped, followed over the years, and then mapped and explained it to us in a very fascinating manner. Thank you.
Thank you. And again, I am saying that thank you very much, Professor Sukumar and Dr. Pratiti, for giving this opportunity. And again, I'm saying that I'm privileged that I'm the first speaker of this series. Thank you again.
But sir, I have do you engineer enzyme in your lab? Means experimentally? Do you?
So basically, we are not doing any, I will say in vitro or the wet lab, but I have a collaborator in TFR. So
Okay,
suppose that
okay.
if we are proposing some type of mutations, they usually check it.
Okay, okay. So I'm taking it as that there is no question from the audience.
Okay.
Dr. Dubey, thank you so much. And it's a very nice talk. Thank you so much.
Okay, welcome. Welcome. Okay, bye for now.
Okay, bye.
Thank you. Bye-bye. Bye.